Welcome to the deep dive. Today, we are really immersing ourselves in what you might call the foundation of therapeutic mastery, the dynamic science of pharmacotherapy.
That's right.
Our sources, they make it incredibly clear that optimal drug therapy, well, it goes far beyond just, giving a pill.
Mm-hmm.
It requires a deep, almost clinical understanding of the drug itself, the disease you're treating, the individual patient, and what the sources call the milieu, the whole context.
That's absolutely the critical mandate for professionals in this field. Our mission today really is to achieve that level of mastery,
Okay.
to move beyond just population-based prescribing and really understand individualized medicine. The core truth, highlighted again and again in our material, is that recommended doses are, well, they have to be based on population data. They cater to an average patient.
But the moment you look at a real person, right, that whole concept of average just kind of starts to fall apart.
Exactly. Few individuals are truly average in how they respond to medicine.
So we need to master these profound sources of variation. Before we jump deeper, though, maybe we should quickly define the language of variation. When we talk about drug action, we always hear pharmacogenetics, PK, and pharmacodynamics, PD.
Yeah, that's a crucial starting point. Pharmacokinetics, PK, it's essentially what the body does to the drug, how it absorbs it, distributes it around, metabolizes it, and then gets rid of it, excretion.
Okay, body on drug.
Right. And pharmacodynamics, PD, that's the flip side, what the drug does to the body. Its mechanism of action, how well it works, its efficacy, and of course, its side effects.
Got it. Drug on body.
And both PK and PD, they are subject to just massive individual variability. Mastering how those parameters shift, with age or disease or genetics, that's really the only path to optimal safe therapy.
Okay, let's unpack this then, starting with the absolute fundamental, defining the dose itself. So at its most basic level, a dose is defined as, quote, the appropriate amount of a drug needed to produce a certain degree of response in a given patient. But the crucial caveat here, the detail that really separates maybe a novice from a master practitioner, is that the dose must be qualified by the specific chosen response you're aiming for.
This distinction, it defines therapeutic intent. You really can't just prescribe the dose of aspirin, for example.
Right.
You have to prescribe the dose of aspirin needed for a very specific outcome. Our sources give us a perfect example, actually a triple example of this.
Okay, let's hear it.
So if you're treating just a simple headache, you prescribe the analgesic dose. That's about 0.3 to 0.6 grams. That amount achieves pain relief at the target site.
Okay, standard headache dose.
Yeah, but if your goal shifts completely, say, to preventing blood clots, you want an antiplatelet effect, the required dose just plummets. It drops dramatically to a tiny 60 to 150 milligrams per day.
Wow, that's a huge difference. Milligrams versus grams.
Huge. And then if the goal changes again, maybe you're tackling the widespread inflammation of rheumatoid arthritis, the dose has to be massive. It skyrockets to 3 to 5 grams per day.
So, same drug, 3 totally different concentrations needed.
Exactly. 3 widely different doses based purely on the pharmacological goal. And this variability really proves why just using a standard fixed dose is often, well, insufficient.
Okay, so if the standard dose isn't always enough, what strategies do clinicians actually use to tailor therapy when we know a patient might fall outside that average norm? Our sources lay out 4 distinct strategies for individualizing the dose.
Right. We start with the simplest one, the standard dose. This is what you use when the individual variation in PK or PD is pretty minimal.
Okay.
Or, maybe more commonly, when the drug just has a very wide safety margin, a large therapeutic window, we call it.
Right, lots of room for error.
Exactly. Think of things like oral contraceptives or many types of penicillin. You basically give the same dose to almost everyone because that slight variation in response isn't going to compromise safety or how well it works.
Makes sense. What's next?
Moving up in complexity, we have the regulated dose. This applies when the drug affects a physiological function that you can easily and reliably measure. So the dose is adjusted by repeatedly monitoring that physiological outcome itself.
Like blood pressure meds.
Antihypertensives are the classic example, yeah. You look at the patient's blood pressure, that's the regulated function, and you adjust the dose up or down until the blood pressure is where you want it, in the target range.
Mm-hmm.
And what's important to note here, really, is that in this situation, monitoring the actual plasma concentration of the drug usually isn't necessary. The effect itself is your best guide.
Okay, but that changes with the third strategy, doesn't it? The target level dose.
It does. Here, the clinical responses may be difficult or impractical to measure directly, but we've established a strong link. a correlation between therapeutic success and a specific range of the drug's concentration in the plasma.
So this is where drug-level monitoring becomes essential.
Absolutely mandatory, yes. This is critical for drugs with a narrow therapeutic index where the gap between effective and toxic doses is small, or where the clinical effects take a long time to show up. Think antidepressants, some antiepileptics, digoxin, theophylline. You start with an initial sort of empirical dose, and then you check the plasma concentration against that known target range. Subsequent adjustments are then based on the measured level, not necessarily the immediate clinical effect you're seeing.
Got it. And the last one, the titrated dose. This sounds like maybe the trickiest one.
It can be, yeah. It arguably involves a therapeutic compromise. The clinician is aiming for the maximum possible therapeutic effect, while simultaneously making sure the adverse effects, the side effects, remain tolerable for the patient.
So finding a balance.
Exactly. Titration is really common with drugs that have inherently high toxicity, things like anti-cancer drugs or levodopa for Parkinson's disease. Now, if the drug is for a non-critical situation, you typically titrate low to up. You increase the dose cautiously until the side effects become the limiting factor.
Okay.
But in critical care, say, you're treating a life-threatening arrhythmia, you might actually start high to get immediate control and then carefully titrate down to find the lowest effective, acceptable maintenance level. The goal is always finding that optimal balance point.
This inherent complexity, this need for adjustment, it leads us right into the debate around fixed-dose combinations, or FDCs. Putting two or more drugs together in a fixed ratio in just one pill or capsule. These are everywhere.
They really are ubiquitous, and our sources dive deep into their pros and cons. FDCs are often championed, mainly for convenience and, at least in theory, better patient compliance.
Sure, one pill is easier than two or three.
Right. If a patient genuinely needs all the components, giving them in a single pill definitely streamlines their daily routine. They can also sometimes offer cost savings in manufacturing and packaging.
And beyond just convenience, there are some strong therapeutic arguments for them too, right?
Oh, absolutely. The clearest one is synergy. The sources cite that classic example, sulfamethoxazole plus trimethoprim.
Bactrim or Septra.
Exactly. That combination's effectiveness is way more than just the sum of its parts because they block two sequential steps in how bacteria make folate.
Okay, that makes sense.
Yeah.
What else?
Another really crucial advantage is in fighting drug resistance, especially in chronic infections. Combining drugs ensures that a single drug isn't given alone, which is vital in treating things like tuberculosis, HIV/AIDS, and falciparum malaria, where using just one drug, monotherapy, quickly leads to resistance developing.
Hitting it from multiple angles.
Precisely. And they can also combine drugs that have additive therapeutic effects but maybe different side effect profiles. Like combining amlodipine and atenolol for hypertension, you get additive blood pressure lowering but potentially fewer side effects than using a high dose of either one alone.
Okay, those are some compelling advantages. But the sources, they seem pretty uncompromising about the downsides, the inbuilt disadvantages, which they argue often outweigh the benefits.
They do. And the biggest clinical limitation, the fundamental problem, is the complete inability to individualize the dose of the components.
Right. It's fixed.
It's fixed. So imagine you have a patient with, say, mild kidney problems, and one of the drugs in the FDC is cleared by the kidneys and really needs a dose reduction. You simply cannot adjust that one drug down without automatically lowering the dose of the other component too, which might not need any adjustment at all. It completely undermines that core principle of individualized prescribing we just talked about.
Yeah, you lose all flexibility.
You do. Furthermore, the different components might have wildly different durations of action, their time course. So taking them together at a single fixed interval might be totally inappropriate pharmacologically for one or both drugs.
I hadn't thought of that.
And then, practically, when a patient taking an FDC reports an adverse reaction, it becomes extremely difficult, often impossible, to say definitively which component caused it.
Right. Which drug is the culprit?
Exactly. And the broader issue, really, is that FDCs are often promoted very vigorously by manufacturers, sometimes based on pretty minimal scientific rationale. This leads to market confusion and, frankly, the fundamental irrationality of giving a patient a drug they simply don't need, exposing them to unnecessary side effects and expense.
And this isn't just a theoretical problem, is it? The sources point out this issue is significant enough to require actual global regulatory action.
It really is. The World Health Organization's essential medicines list, it only includes about 25 FDCs. And India's national list of essential medicines has only 20.
That's a tiny number compared to what's out there.
It is. And this is against the backdrop of that massive ban. The sources highlight the recent prohibition of 344 irrational FDCs in India. That really underscores just how widespread this poorly justified prescribing practice had become. So, with those fundamentals of dosing established, let's shift the focus now squarely onto the patient. How do individual patient factors modify drug action? It can happen in two main ways. The drug's effect can be quantitatively altered.
Meaning just more or less effect.
Exactly. The action is increased or decreased, and we usually deal with that by adjusting the dose like we've discussed.
Okay.
Or, and this is far more dangerous, the action can be qualitatively altered. This leads to things like idiosyncratic reactions or allergies.
Things that aren't just dose-related.
Right. And these often require stopping the drug permanently.
Got it. So starting with the quantitative factors, one of the most obvious adjustments needed is based on body size, right? Standard adult doses are historically based on, what, a 70 kilogram?
Yeah, roughly a 70-kilogram medium build. But obviously, patients come in all sizes. So for those at the extremes, very obese or very lean, simple dose calculation based on body weight, VW is pretty common.
Just scale it up or down by weight.
Sort of, but the research actually suggests a more nuanced approach might be better. Body surface area, or BSA, is often argued to be a more physiologically accurate measure.
Why BSA?
Because it tends to correlate better with things like metabolic activity, total body water, and extracellular fluid volume factors that really influence how a drug is distributed and eliminated.
That makes perfect sense. Metabolism is key for drug clearance, as you said, yet BSA-based dosing sounds
Well,
cumbersome to calculate in a busy clinic.
it absolutely is. That's the trade-off. Although BSA probably provides a superior index, and it is used rigorously for highly toxic compounds like anti-cancer drugs where precision is paramount. For most routine prescribing, body weight remains the standard index. Or often we rely on manufacturer recommendations that are specified directly as milligrams per kilogram.
Okay, now let's talk age. The sources are really emphatic about this. Infants and children are not simply small adults. We absolutely must detail the profound physiological differences that create unique risks.
This is critical. We have to start with elimination, drug removal, which is just immature in a newborn. They have a low glomerular filtration rate. The GFR in their kidneys is low, and their tubular transport mechanisms, which also help remove drugs, are immature.
So drugs hang around longer.
Much longer. This drastically prolongs the half-life, the drugs that are primarily cleared by the kidneys, like gentamicin or penicillin. This prolonged elimination puts them at a really high risk for cumulative toxicity.
And how long does that immaturity last?
Well, we see that GFR doesn't really reach adult levels until about 5 months of age. And those tubular secretion mechanisms, they take even longer, maybe around 7 months to fully mature.
Wow, okay. What about the liver?
The hepatic system is also underdeveloped. They have inadequate activity of the conjugation and detoxifying enzymes that break down drugs. This leads to some tragic, specific syndromes.
Like gray baby syndrome.
That's the classic example, yes. With the antibiotic chloramphenicol, the immature liver simply cannot metabolize the drug effectively, leading to accumulation and that devastating syndrome.
And the brain is more vulnerable too.
Yes. Adding to the complexity, their central nervous system is more vulnerable. The blood-brain barrier, which normally protects the brain, is more permeable in infants, allowing greater drug penetration.
Okay.
This is why unconjugated bilirubin, which is usually processed by the liver, can easily cross that leaky barrier in neonates and cause kernicterus, a severe type of brain damage.
So kidneys, liver, brain barrier. Even absorption is different.
Right. It is. They tend to have lower gastric acidity and slower intestinal transit times, which can sometimes delay or reduce oral drug absorption.
But faster through the skin.
Exactly. Conversely, their skin is very thin and permeable. This means they exhibit much faster and more significant transdermal absorption. Clinicians have to be acutely aware of this when prescribing any topical creams or ointments.
Okay, but then the physiology shifts dramatically after infancy.
It really does. Post-infancy, children often metabolize drugs faster than adults do.
Faster. Why?
The rate of their hepatic microsomal enzyme activity actually becomes elevated compared to adults for a period. This means that for drugs like theophylline, phenytoin, or even digoxin, the half-life is shorter.
So they might need a higher dose per kilo.
Often, yes. A higher dose per kilogram than the adult standard might be needed just to maintain therapeutic concentrations. It's counterintuitive, but crucial.
And we also have to catalog those special, sometimes irreversible adverse effects that are unique to growing children.
Absolutely. Corticosteroids, if used long-term, can suppress growth. Androgens, used inappropriately, can cause permanent stunting.
And the teeth issue with tetracyclines.
Yes, tetracyclines, a common class of antibiotics, cause permanent discoloration and potential deformity of developing teeth and bones. These effects are unique to that developmental phase.
Okay, shifting gears to the other end of the age spectrum, the elderly, a population highly prone to cumulative toxicity. Why is that?
It's largely due to age-related decline in organ function, plus the very common issue of polypharmacy taking multiple drugs.
Right. What's the primary organ compromise?
It's usually renal function, the kidneys. Our sources state that GFR, that measure of kidney filtration, is typically reduced by about 25% by age 50 and down by 50% by age 75.
50%, that's a huge decline.
It's relentless, and it is probably the single most important factor determining dose adjustment in the elderly.
So dose reduction is basically mandatory for kidney-cleared drugs.
Pretty much. Significant dose reduction is needed. For instance, the sources detail a specific guideline for the antibiotic streptomycin. The standard daily dose of 1 gram should be cut to 0.75 grams after age 50, and then further reduced to just 0.5 grams after age 70. This kind of proportional reduction based on estimated GFR is absolutely essential.
Okay. What about beyond the kidneys? Liver function.
That often declines too. They frequently experience reduced hepatic microsomal enzyme activity and also decreased blood flow to the liver. Absorption from the gut can be slower due to reduced motility. And critically, many elderly patients have lower levels of plasma albumin, the main protein that binds drugs in the blood.
And less protein binding means more free drug.
Exactly. When protein binding is reduced, more of the drug remains unbound and pharmacologically active. This increases the potential for toxicity, especially with drugs that are normally highly protein-bound, like the anticoagulant warfarin.
Right. More free warfarin means higher risk of bleeding.
Precisely.
Okay.
And on top of these PK changes, the pharmacodynamics, how the body responds, can change too. Beta-adrenergic receptors, for example, often show reduced responsiveness, and there's an increased sensitivity to anticholinergic side effects.
Like dry mouth, constipation.
Yes, and even conversely, other drugs like the antifungal ketoconazole, metoclopramide for nausea, or even digitalis can sometimes induce gynecomastia breast enlargement, which is obviously a side effect exclusive to male patients.
Interesting. And pregnancy. Especially late pregnancy. That must really complicate things.
Oh, massively. The third trimester, in particular, introduces huge physiological changes that fundamentally alter how the body handles drugs, drug disposition.
How so?
Well, the changes are complex and can even seem contradictory. Reduced gastrointestinal motility might delay absorption.
Okay.
But then you have expanded plasma volume and extracellular fluid, which increases the volume of distribution, VOD, for many water-soluble drugs.
So they spread out more.
Right. But at the same time, renal blood flow is markedly increased, which accelerates the elimination of polar water-soluble drugs. And sometimes liver enzymes involved in metabolism are induced, working faster.
So it's a real mixed bag.
It is. Clinicians have to account for this really variable drug disposition to make sure they aren't accidentally underdosing the mother or, crucially, compromising fetal safety.
Okay. Finally, in this section, we have to acknowledge external factors. The route of administration, the environment, and timing. Route seems obvious. How you give the drug matters.
Absolutely. The chosen route governs the speed and intensity of the drug's action. Parenteral routes injections are almost always more rapid and predictable than taking something orally.
And the route can completely change what the drug is used for.
Completely. Magnesium sulfate is a fantastic example from the sources. Give it orally, it acts as a purgative, a laxative.
Okay.
Apply it topically as a paste or compress, it helps reduce swelling and sprains.
Right.
But give it intravenously, IV, and it acts as a central nervous system depressant and can lower blood pressure. Same molecule, totally different uses, dictated purely by the route.
Wow. And environmental factors, like pollution or diet.
They can have subtle but measurable impacts, yeah. Exposure to certain environmental toxins like chemicals in tobacco smoke or even compounds formed when you charcoal broil meat can actually induce hepatic drug metabolizing enzymes, make them work faster.
Meaning you might need a higher dose of some drugs.
Exactly. You might need higher doses for certain drugs to achieve the same effect in someone who's regularly exposed to these inducers.
And this brings us to timing chronopharmacology. When you take the dose, matters.
It absolutely matters because our body's physiology operates on natural circadian rhythms, 24-hour cycles. Statins, for example, which inhibit cholesterol synthesis. They are generally most effective when taken in the late evening. Why? Because that aligns with the body's natural peak time for cholesterol production overnight.
Clever. Any other examples?
Yeah, corticosteroids. Taking the entire daily dose as a single dose in the morning helps minimize the disruption of the body's natural pituitary adrenal axis, that natural cortisol rhythm. This reduces the risk of suppressing the body's own steroid production.
Okay, now let's explore how the mind, our psychology, and major disease states interact with medicine. We have to start with the psychological factors. The power of belief, really, embodied by the placebo and its opposite, the nocebo effect.
Right. A placebo is technically an inert substance, a sugar pill or saline injection that works purely through psychodynamics, basically, the belief system and expectations of the patient.
But it can have real effects, right?
Oh, absolutely demonstrable effects, sometimes quite profound physiological responses. The most compelling example is probably placebo analgesia pain relief from a placebo.
How does that work?
It seems to work, at least in part, by stimulating the release of the body's own natural painkillers, endogenous opioid peptides, or endorphins.
And the proof is that you can block it.
That's the really fascinating part, noted in the sources, yeah. This placebo-induced pain relief can be entirely blocked by giving naloxone, which is an opioid antagonist drug. It stops the endorphins from working.
Wow, so it's a real chemical effect triggered by belief.
It seems so, but the key clinical issue is that the placebo effect is highly variable. It can vary even within the same person on different days. This variability really limits its use in practical therapeutics, although it remains absolutely essential as a control treatment in clinical trials.
And the nocebo effect is just the opposite.
Exactly, and the converse. Negative expectations, pessimism, maybe loss of faith in the treatment, these can actively oppose the therapeutic action of an otherwise effective drug, a negative psychodynamic effect.
Okay, but the major clinical concern, usually, is the direct physical impact of existing diseases, pathological states, starting with the GI tract.
Yes, gastrointestinal diseases can really complexly alter drug absorption.
How so?
Well, conditions like coeliac disease can drastically change absorption characteristics. For example, the sources mention that absorption of the antibiotic amoxicillin might be decreased in coeliac patients, while absorption of another antibiotic, cephalexin, might actually be increased. It's not always predictable. And other mechanical issues like gastric stasis, just slow emptying of the stomach, which can happen in conditions like diabetic gastroparesis or even during a migraine attack, can significantly retard or delay drug absorption.
And stomach acid levels.
That matters too. Achlorhydria, having low or no stomach acid, can decrease the absorption of weakly acidic drugs like aspirin, which need an acidic environment to be absorbed well.
Okay. Moving to major organ failures. We absolutely have to dive deep into liver disease, specifically cirrhosis. PK and PD are fundamentally compromised here, right?
Fundamentally, yes. Liver failure impacts drugs in multiple, often interacting ways. First, reduced function of the liver cells, the hepatocytes, means you lose that crucial first-pass metabolism.
The initial breakdown as the drug passes through the liver after absorption.
Exactly. So for drugs that are normally extensively metabolized on that first pass, drugs like morphine, propranolol, lidocaine, their bioavailability, the amount reaching the bloodstream increases dramatically.
Meaning you need lower doses.
Significantly lower doses are absolutely mandated to avoid toxicity.
Okay, what's the second impact?
Second, the diseased liver synthesizes fewer plasma proteins, particularly albumin. Reduced serum albumin means less capacity to bind drugs.
Right, we mentioned this with the elderly.
Same principle. It leads to a higher concentration of the free, active drug circulating in the blood. This is especially dangerous for acidic drugs that are normally highly protein-bound, like warfarin. The risk of bleeding just skyrockets.
Got it. And third?
Third, there's just decreased metabolic capacity overall. The remaining liver enzymes can't keep up. This means we have to reduce the maintenance dose of drugs metabolized by the liver, like morphine and lidocaine again, to prevent them from accumulating to toxic levels.
And avoid prodrugs.
Yes, clinicians absolutely must avoid prodrugs, drugs that are inactive until the liver activates them. Things like vacampicillin, an ampicillin prodrug. They might become ineffective if the liver can't perform that activation step.
And the most dangerous change in liver disease.
Arguably, it's the increased sensitivity of the central nervous system. Patients with cirrhosis are exquisitely sensitive to CNS depressant drugs.
like sedatives or painkillers.
Exactly. Normal therapeutic doses of morphine or barbiturates can easily push these patients into hepatic encephalopathy, basically a state of confusion, disorientation, and potentially coma because the damaged liver can't clear ammonia and other toxins that affect the brain.
Okay, equally critical is kidney disease, specifically renal failure. This presents a huge dosing challenge too.
Absolutely. The clearance, the removal of drugs that are excreted largely unchanged by the kidneys. Classic examples are aminoglycoside antibiotics like gentamicin, digoxin for heart failure, phenobarbital. Their clearance is reduced almost in direct proportion to the decrease in the patient's creatinine clearance, CLCR, which is our main measure of kidney function.
So dose adjustment is directly tied to CLCR.
It has to be precise. The loading dose, the initial dose to quickly reach therapeutic levels, generally remains the same. But the maintenance dose, the ongoing dose, must be proportionally reduced based on that CLCR value.
Are there specific guidelines, thresholds professionals use?
Yes, the source material provides clear boundaries that are essential knowledge. If the patient's CLCR is between 30 and 50 milliliters per minute, the dose rate, how much drug over time, must be reduced to about 50% of the normal rate.
Cut it in half.
Essentially, yes. And if the CLCR drops even lower, say between 10 and 30 milliliter, the dose must be cut further down to about 30% of normal. This is absolutely essential to prevent severe, sometimes life-threatening cumulative toxicity, especially with drugs like aminoglycosides that have narrow safety margins.
And beyond just impaired clearance, kidney failure also increases sensitivity to some drug effects, particularly in the brain.
Yes, that's another key point. The permeability of the blood-brain barrier seems to increase in renal failure. This means patients can experience greater CNS depression from standard doses of sedatives and opioid painkillers.
Is that why certain drugs are completely avoided?
It is. This is why the sources strictly advise avoiding the opioid pethidine, also known as meperidine, in renal failure. Its metabolite, norpecadine, which is actually excitatory to the CNS, accumulates because it can't be cleared by the failing kidneys.
And that accumulation causes.
It can lead to CNS hyperexcitability, tremors, muscle twitches, and even severe seizures. Even at what seem like normal therapeutic doses of pethidine, it's very dangerous.
What other drugs need caution in renal failure?
Other drugs to use with extreme caution or avoid include NSAIDs like ibuprofen because they can worsen fluid retention and further impair kidney function. Also, potassium-sparing diuretics are risky due to the high danger of hyperkalemia, excessive potassium levels.
Okay, let's consider congestive heart failure, CHF. Here, even if the kidneys and liver are structurally okay, their function is compromised because the heart isn't pumping effectively, right? So perfusion is down.
Exactly. Reduced cardiac output is the core problem in CHF, and it has downstream effects. It leads to decreased drug absorption from the gut, partly due to edema, swelling in the gut wall, and also due to vasoconstriction in the splanchnic circulation, the blood vessels supplying the gut.
Okay, absorption is down. What else?
The volume of distribution, VOD, can be altered. Often, lower loading doses are required for drugs like lidocaine, used for arrhythmias. But perhaps most importantly, elimination is retarded.
Why?
Because blood flow to both the liver and the kidneys is reduced due to the poor cardiac output. This prolongs the half-life of most drugs that rely on these organs for clearance. And on top of that, a decompensated, failing heart muscle often shows increased sensitivity to the effects, both good and bad, of digitalis drugs like digoxin, increasing the risk of cardiotoxicity.
Okay, finally, we need to touch on other diseases that cause altered pharmacological sensitivity, not just altered PK. Thyroid disease is a prime example mentioned.
Yes, it's a really good example of altered PD. Patients who are hypothyroid with low thyroid function are generally more sensitive to CNS depressants like morphine and also more sensitive to digoxin.
So they need lower doses.
Often, yes. Conversely, patients who are hyperthyroid with an overactive thyroid tend to be resistant to digoxin's desired inotropic action, its ability to strengthen the heart's contraction.
But more prone to side effects.
Paradoxically, yes. While resistant to the therapeutic effect, they are often more prone to digoxin's arrhythmogenic side effects, its tendency to cause abnormal heart rhythms. This difference in pharmacodynamic response is really crucial for clinicians to know.
And there were other specific sensitivities noted in the source material, too.
Yes, a few important ones. Patients who've recently had a myocardial infarction, a heart attack, are known to be more prone to arrhythmias caused by sympathomimetic drugs like adrenaline or by digitalis. Patients with myasthenia gravis, that muscle weakness disorder, are extremely sensitive to muscle relaxants like curare, and related drugs used in anesthesia. Even small doses can cause profound paralysis.
Wow.
And interestingly, on the other side of the coin, patients with schizophrenia often seem to tolerate unusually large doses of phenothiazine antipsychotic drugs without showing the significant sedation or adverse effects you'd expect. These are all highly specific pharmacodynamic variations based on the underlying pathology.
Okay, now we enter the fascinating realm of genetics. Genetics, the source's state, controls essentially every single key determinant of how we respond to drugs.
That's right. It controls the enzymes that metabolize drugs, the transporters that move drugs across cell membranes, and the receptors and ion channels that are the ultimate targets of drug action. This inherent genetic variability is arguably the greatest frontier in customizing therapy.
We hear two terms often, pharmacogenetics and pharmacogenomics. What's the difference?
It's subtle but useful. Pharmacogenetics traditionally focuses on how variation in one or just a few specific genes influences drug response. Think of it as the study of the genetic basis for individual variation.
Okay. And pharmacogenomics?
Pharmacogenomics is broader. It refers to the use of information about the entire human genome, all the genetic material, to guide the choice of drug and the dose for an individual patient. The shared goal, though, is personalized medicine.
Right. The idea is to identify upfront who's likely to benefit from a drug, who's likely to fail, and maybe most importantly, who's at high risk of suffering severe toxicity.
Exactly.
Now, while most drug responses show continuous variation like a bell curve distribution of the population, some genetic defects cause what's called discontinuous variation. It's more like an all-or-nothing response rooted in a single specific gene defect.
Yes, and the classic examples are absolutely required knowledge for anyone in this field. First is atypical pseudocholinesterase.
What happens there?
It's a defect in the enzyme that normally breaks down succinylcholine, a very common muscle relaxant used during surgery and intubation. If a patient has this atypical enzyme, they can't break the drug down quickly.
So the paralysis lasts much longer.
Far longer than expected, yeah. What should be a few minutes of paralysis can turn into hours, requiring prolonged mechanical ventilation. It's a dramatic effect from a single gene defect.
Okay. What's the other key example?
The other really key one is G6PD deficiency. That's glucose 6-phosphate dehydrogenase deficiency. It's an X-linked monogenic trait, meaning it's carried on the X chromosome, and it's surprisingly common in certain populations.
Which populations?
Particularly common in people with Mediterranean, African, or Southeast Asian ancestry.
And what's the danger with G6PD deficiency?
The danger lies in causing rapid and severe hemolysis, the destruction of red blood cells, when the affected person is exposed to certain oxidizing drugs. Their red cells lack the protective enzyme and basically fall apart under oxidative stress.
And there's a list of high-risk drugs.
Yes. The research points out a critical list of high-risk agents that must be used with extreme caution or completely avoided in these individuals. The highest risk drugs include the antimalarial primaquine, nalidixic acid, dapsone, nitrofurantoin, sulfonamides, and methylene blue. This is a critical screening consideration, often based just on ethnic background initially.
Okay, moving beyond these single gene defects, let's discuss the more common gene variations that affect drug metabolism. We were talking about polymorphisms, right? What exactly is that?
Right. A polymorphism generally refers to a variation in the DNA sequence that's present in a significant portion of the population. Often, we're talking about a single nucleotide polymorphism, or SNP, pronounced SNP.
Just a single letter change in the DNA code.
Exactly. A single base pair substitution, like an A changing to a G, that occurs at a frequency of greater than 1% in the population. And these seemingly tiny SNPs can dramatically alter the function of the proteins they code for, especially drug metabolizing enzymes.
Okay, give us some major examples.
A really important one involves the enzyme CYP2C9. Certain common variants, or SNPs, in the CYP2C9 gene cause it to work much more slowly.
And CYP2C9 metabolizes.
Warfarin. It's a key enzyme for metabolizing warfarin. So patients who inherit these slow-functioning variants are poor metabolizers of warfarin.
Meaning a standard dose leads to high levels.
Exactly. A standard dose results in excessively high drug concentrations, leading directly to a significantly increased dangerous risk of bleeding. Dose reduction based on their genotype is absolutely mandatory for safety.
Okay. Another key example related to severe toxicity.
TPMT deficiency. That's thiopurine methyltransferase deficiency. This enzyme metabolizes important immunosuppressive and anti-cancer drugs like 6-mercaptopurine and azathioprine.
And deficiency causes.
If you lack functional TKMT, you can't break these drugs down properly. Standard doses lead to massive accumulation and a very high risk of severe, potentially fatal bone marrow toxicity. Genetic testing before starting these drugs is now standard practice in many places.
Wow. Another one mentioned is UGT1A1.
Yes, the UGT1A128 allele. This variant is linked to an increased risk of severe neutropenia, low white blood cells, and severe diarrhea when patients are treated with the chemotherapy drug irinotecan. Again, knowing the genotype allows for dose adjustment or closer monitoring to prevent these catastrophic side effects.
There's also a very familiar clinical polymorphism involving N-acetyltransferase 2, or NAT2, right? This determines acetylator status.
That's right. The NAT2 enzyme is responsible for acetylation, a key metabolic pathway for several drugs. Genetic variations in NAT2 divide the population roughly into rapid acetylators and slow acetylators.
And slow acetylators are a risk.
Yes. Slow acetylators metabolize these drugs poorly and tend to accumulate them. They are prone to specific toxicities like peripheral neuropathy from the tuberculosis drug isoniazid or the drug-induced lupus syndrome sometimes seen with hydralazine or percanamide.
So you might need lower doses or co-treatments?
Exactly. For instance, slow acetylators taking isoniazid are usually given vitamin B6 concurrently to help prevent the neuropathy.
Okay, and perhaps the most widely impactful example, showing both toxicity and therapeutic failure, is the CYP2D6 abnormality.
Yes, CYP2D6 is a hugely important enzyme. It metabolizes something like 25% of all commonly used drugs. Genetic defects leading to poor CYP2D6 function create a poor metabolizer status, which is found in about 5–10% of Caucasian populations, for example.
And the consequences?
Two main consequences. First, these patients experience toxicity with standard doses of many drugs that are CYP2D6 substrates. Think certain antidepressants, antipsychotics, beta blockers like metoprolol. The drug builds up.
Okay, increased toxicity risk.
But second, and this is fascinating, it can cause complete therapeutic failure for certain prodrugs. The best example is codeine.
Codeine needs CYP2D6.
Right, yes. Codeine itself is not very active and must be converted into its active form, morphine, by the CYP2D6 enzyme in the liver to provide pain relief.
So in poor metabolizers?
In poor metabolizers, that conversion doesn't happen effectively. Codeine completely fails to produce analgesia. They get no pain relief from it, no matter the dose. It highlights how genetics can wipe out efficacy entirely.
And genetic variability doesn't just affect metabolism, right? It can affect the drug's target.
Absolutely. Malignant hyperthermia is the classic example here. This isn't an enzyme issue. It's a genetic defect in the drug's target. Specifically, the ryanodine receptor, which is a calcium release channel in skeletal muscle.
Okay.
In susceptible individuals, this channel is abnormal. When triggered by certain general anesthetics, particularly halothane, or by succinylcholine again, the channel opens uncontrollably, releasing massive amounts of calcium into the muscle cells, leading to intense muscle rigidity, a massive increase in metabolic rate, and a catastrophic, often fatal rise in body temperature malignant hyperthermia. It's a pharmacogenetic emergency.
So the foundational premise of pharmacogenomics seems really appealing, as you said. Genotype someone once, and you have that information for life.
That's the promise, yes.
But what's the reality? How widely is it used now?
Currently, its widespread practical application is still somewhat limited. The main barrier has been the need for multiple, often drug-specific genotypic tests. However, simple, cost-effective point-of-care tests for high-impact conditions like G6PD deficiency are already becoming quite routine in certain settings. The field is moving fast, though.
Okay. Considering this vast complexity, dose definitions, patient factors, genetics, how do we actually ensure we practice medicine rationally? The World Health Organization, the WHO, provides a really good definition.
They do. Their definition of rational use is that patients receive medication appropriate to their clinical needs in doses that meet their own individual requirements for an adequate period of time and, importantly, at the lowest cost to them and their community.
That sounds like a high bar. What does rational prescribing look like in practice? What are the criteria?
It really involves a mental checklist for every prescription. First, is there an appropriate indication for using a drug at all?
Okay, step one. Is it needed?
Exactly. Second, if yes, have I selected the appropriate drug? Considering its efficacy, its safety profile, and its suitability for this specific patient.
Right.
Third, is the dose, the route, and the duration of treatment correct? Fourth, Are there any contraindications or major interactions I need to worry about?
Hmm.
Fifth, will the dispensing be correct? And will the patient receive clear instructions they can understand and follow? And sixth, critically, is there a plan for adequate monitoring of adherence, therapeutic effect, and potential adverse effects?
Yeah, it's a comprehensive checklist.
It needs to be.
But here's where it gets really interesting and maybe a bit concerning. Focusing on the common mistakes. Despite these clear criteria, irrationality in prescribing remains, well, rampant according to our sources. They give some excellent examples of these common pitfalls.
Yes, unfortunately they do. One of the most pervasive mistakes is simply using a drug when none is needed at all.
Like antibiotics for a cold.
That's the classic example, yes. Prescribing antibiotics for a straightforward viral fever or upper respiratory infection where they have zero benefit but contribute to resistance. Another common one is the compulsive co-prescription of things like vitamins or tonics or nutritional supplements alongside other medicines, even when the patient has no deficiency and gets no real benefit.
Okay. What about selecting the wrong drug?
That happens too, often based on poor patient assessment or just not considering the drug's full profile. For instance, prescribing a beta blocker, a common blood pressure medicine, to a patient for their hypertension, but failing to note they also suffer from asthma, where beta blockers are generally contraindicated because they can worsen breathing.
A dangerous oversight.
Or using really broad-spectrum, powerful antibiotics like ciprofloxacin for something simple like a common sore throat, maybe pharyngitis, which is usually viral and needs no antibiotic anyway. Or perhaps just a simple penicillin if it were bacterial. It's using a sledgehammer to crack a nut, and it drives resistance.
We also see the use of drugs with questionable effectiveness.
Absolutely. The sources mention things like many traditional cough mixtures, where the evidence for significant benefit over placebo is often weak. Or the use of enzymes like serratiopeptidase for reducing swelling, which again lacks rigorous, high-quality evidence supporting its widespread use.
And maybe the most dangerous irrationality mentioned is polypharmacy.
Unnecessary polypharmacy, yes. This is where a patient ends up taking multiple drugs, sometimes 5, 10, even 12 or more medications concurrently.
Right.
Often, some of these drugs are treating the side effects of other drugs on the list. It exponentially increases the risk of complex, unpredictable, and potentially dangerous adverse drug reactions and interactions.
And the consequences of all this irrationality?
They're severe. Increased morbidity, more illness, and even mortality. Significant financial loss, both for the patient and the healthcare system, loss of patient confidence in the medical profession, and, maybe most critical from a public health perspective, the relentless emergence and spread of antimicrobial resistance, driven largely by the overuse and misuse of antibiotics.
So how do we combat these pitfalls, this reliance on maybe just habit or anecdote? This is where evidence-based medicine, EBM, comes in.
Exactly. EDM represents a fundamental shift in clinical decision-making. It's a move away from relying solely on personal experience or authority, experience-based medicine, towards decisions based on conscientiously using the best available, systematically evaluated research findings.
And the bedrock of EBM is the clinical trial, right?
Yes, the well-designed clinical trial. It's defined as a prospective, meaning planned in advance, ethically designed investigation intended to objectively compare different therapeutic measures, like a new drug versus a placebo or an existing standard treatment.
And for these trials to be credible, they have to follow strict standards. GCP.
Yes, they must adhere to rigorous international ethical and scientific quality standards known as Good Clinical Practice, or GCP. And underpinning GCP are those 4 core ethical pillars we must always consider.
Remind us what they are.
They are autonomy, which is the right of the research subject to make their own informed decisions, including the right to withdraw at any time. Beneficence, the obligation of the researchers to maximize the potential benefits of the research. Non-maleficence, the absolute obligation to minimize potential harm or risk to participants. And justice, ensuring the fair distribution of both the risks and potential benefits of the research among different groups. Adherence to these principles ensures the science is both scientifically sound and morally justifiable.
Okay. Now, a core challenge in any trial is controlling for bias, conscious or unconscious. How do we do that?
We achieve bias control primarily through 2 essential methodological techniques: randomization and blinding, or masking.
Explain randomization.
Randomization means that participants are allocated to the different treatment groups, say the new drug group or the placebo group, entirely by chance, like flipping a coin or using a computer algorithm. This prevents the investigators from consciously or unconsciously selecting certain types of patients for one group over another, which would introduce selection bias.
You can even stratify it.
Yes, you can use stratified randomization. Before starting, you might divide patients based on important characteristics like age, sex, or how severe their disease is, and then randomize within each of those strata. This helps ensure the groups are well balanced on these key factors from the outset.
Okay, and blinding.
Blinding, or masking, conceals the nature of the treatment being received. In a single blind trial, only the participant, the subject, is unaware of whether they are receiving the active drug or the placebo control.
Okay.
But in a double-blind trial, neither the participant nor the investigator or research staff who are assessing the outcomes know who is getting which treatment. This prevents bias in both reporting of symptoms by the patient and assessment of outcomes by the researchers.
And the gold standard is putting these together.
Exactly. The ultimate, most credible source of clinical evidence is generally considered to be the randomized controlled double-blind trial, the RCT.
Now, even with good design, trials can make statistical errors, right? Type I and type 2.
Yes, that's a key concept in interpreting trial results. A type I error, often denoted by alpha, is a false positive. It's concluding that there's a difference between treatments say that the new drug is effective when, in reality, there isn't one. The difference observed was just due to chance.
Okay, seeing a difference that isn't real.
Right. A type 2 error, denoted by beta, is the opposite, a false negative. This is failing to detect a genuine difference between treatments that truly exists. The trial might conclude the drug isn't effective when actually it is, but the study just wasn't large enough or sensitive enough to pick it up.
And the power of a study relates to this.
Exactly. The statistical power of a trial, which is one brought by, is its ability to detect a true difference if one exists. When designing an RCT, researchers calculate the required sample size, how many patients they need specifically to ensure the trial has sufficient power, usually 80% or 90%, to minimize the risk of a type 2 error, while also setting a limit, usually 5%, on the acceptable risk of a type I error, the p-value threshold.
Makes sense. Now, what if we have multiple RCTs on the same topic?
That's where the highest level of evidence often comes from. Meta-analysis. This is a statistical technique that systematically combines, or pools, the data from multiple, similarly conducted, high-quality RCTs addressing the same question.
Why pool them?
By pooling the results, you dramatically increase the total number of patients studied, which increases the statistical power and precision. This allows for a much more reliable estimate of the true treatment effect and helps bring out the overall balance of evidence, ironing out inconsistencies between smaller individual trials.
Like the evidence for statins.
Statins in preventing coronary artery disease is a perfect example. Many individual RCTs showed benefits, but it was the large meta-analyses pulling tens or even hundreds of thousands of patients that provided the unassailable, definitive evidence supporting their widespread use.
Okay. Now, not all evidence comes from RCTs. What about observational studies like cohort studies?
Right. Observational studies are important too, especially for questions RCTs can't easily answer, like long-term safety. A cohort study is purely observational. The researchers don't intervene, they just watch. They typically identify a group of people who are using a particular drug, the exposed cohort, and compare their outcomes over time to a similar group of people who are not using the drug, the unexposed cohort.
What are they good for?
They are particularly valuable for studying large populations over long periods and are primarily used to discover relatively uncommon or delayed adverse effects that might have been missed in the shorter, smaller phase 3 pre-marketing trials. They can be prospective, following people forward in time, or retrospective, looking back at existing records.
And case control studies. They work backward.
Yes, a case control study starts with the outcome and looks backward. Researchers identify patients who already have a specific condition or adverse event, the cases, maybe people who developed a rare blood disorder like a granulocytosis. They then compare the past drug exposure history of these cases to that of a carefully matched group of people who do not have the condition, the controls.
Factor to do.
Much faster and more cost-effective than cohort studies, especially for rare outcomes. But the big limitation is that case control studies can only show an association between a drug and an outcome. They cannot definitively prove causality because they are prone to various biases, like recall bias.
So with all these different study types, how do we rank the evidence? Is there a grading system?
Yes, there's a generally accepted hierarchy for grading the strength of evidence. Grade I evidence, the most reliable, comes from meta-analyses or systematic reviews of high-quality RCTs. This should ideally form the basis of clinical decisions whenever available.
Okay, grade I is top tier.
Right. Grade 2 would typically be evidence from at least one well-designed RCT. Grade 3 might be evidence from well-designed non-randomized controlled trials or perhaps from cohort or case control studies. And grade 4 evidence, the least reliable, consists of things like case reports, case series, or just expert opinion or anecdotal experience.
So anecdotes are at the bottom.
They serve mainly as a signal, maybe suggesting something worth investigating with higher grade studies, but they should never be the sole basis for making important therapeutic decisions. Relying on EBM means prioritizing the higher grades of evidence.
So to really understand rational use, we also need to appreciate how drugs come to be in the first place. The drug development process is, well, notoriously long and expensive, isn't it?
Incredibly long and capital intensive, yes.
And
The sources state it often takes well over 10 years, sometimes closer to 15, from initial discovery to getting a drug approved for marketing. And the cost is staggering, often estimated at somewhere between $500 million and well over $1 billion per successful compound that makes it all the way through.
wow, where do new drugs even come from? How does discovery start?
The initial discovery phase has evolved a lot over time. Historically, we relied heavily on natural sources, plants, microorganisms, fungi. They provided the lead compounds for many early medicines, like opium from poppies or penicillin from mold.
We still do that.
We still screen natural sources, yes,
Hmm.
and often modify natural compounds to improve them. But today, we increasingly employ a more rational approach. This involves using our detailed knowledge of physiology and disease pathology to identify specific molecular targets, maybe a key enzyme or receptor involved in a disease process.
Like the proton pump for acid reflux drugs.
Exactly like the ANCA plus K plus ATPase enzyme, the proton pump, which became the target for drugs like omeprazole. Once you identify a novel target, you can then try to design or find a molecule that interacts with it specifically.
And modern technology must play a huge role now.
Absolutely. Molecular modeling uses powerful computers and knowledge of protein structures to actually design drug molecules that are tailor-made to fit perfectly into the binding site of a target receptor or enzyme, like designing a key for a specific lock.
Mm.
Combinatorial chemistry uses automated robotic systems for high-throughput screening, HTS. This allows chemists to rapidly synthesize and test tens or even hundreds of thousands of different chemical compounds against a target, dramatically speeding up the search for initial hits.
In biotechnology.
Biotechnology, particularly recombinant DNA technology, has revolutionized things. It allows us to produce large quantities of complex human-identical proteins like insulin for diabetes, growth hormone, or highly specific monoclonal antibodies designed to target cancer cells or inflammatory mediators. These biologics are a huge part of the modern drug pipeline.
There's also a point about single enantiomers. What's that about?
Ah, yes, that's an important refinement, driven partly by regulators like the FDA. Many drug molecules are chiral, meaning they exist in two forms that are mirror images of each other, like your left and right hands. These are called enantiomers.
Oh.
Often, only one of the enantiomers is responsible for the desired pharmacological activity. The other mirror image enantiomer might be inactive, or worse, it could contribute to side effects or even antagonize, block the effect of the active one.
It's better to just have the active one.
Generally, yes.
Girl.
There's been significant regulatory pressure and scientific rationale to develop and market drugs as single, pure enantiomers, rather than the traditional racemic mix, a 50-50 mixture of both. This is why, for example, omeprazole, Prilosec, which was a racemic mix, was largely replaced by its single S enantiomer, esomeprazole, Nexium. The idea is potentially better efficacy and fewer side effects.
Okay, so once a promising compound is identified, before it ever touches a human, it has to go through extensive preclinical studies, right? In animals.
Absolutely extensive studies, yes. And these must be conducted according to strict standards known as good laboratory practices, or GLP, to ensure data quality and reliability. This preclinical phase involves initial screening for the desired activity, defining the drug's systemic pharmacology, what it does to various organ systems in animals, characterizing the animal pharmacogenetics, how the animal body handles the drug, and most importantly, conducting detailed toxicity tests.
What kind of toxicity tests?
They are really comprehensive. They include acute toxicity studies, often to determine the LD50, the dose lethal to 50% of animals, which gives a rough idea of the immediate danger level. Then there are are subacute and chronic toxicity tests, lasting from weeks up to 6 or even 12 months, usually requiring testing in at least 2 species, typically one rodent, like a rat, and one non-rodent, like a dog or monkey, to identify potential long-term organ damage and look for species-specific toxicities.
And tests for reproduction? Cancer.
Yes, specific tests are mandatory to assess effects on reproduction and potential to cause birth defects, teratogenicity. Mutagenicity tests look for potential to damage DNA, often using the Ames test, which uses bacteria as a quick screen. And finally, carcinogenicity studies are often required, especially for drugs intended for long-term use. These involve giving the drug to animals for their entire lifespan, basically, to see if it causes tumors.
That's a massive hurdle to clear even before human trials.
It really is. Only a tiny fraction of compounds that enter preclinical testing ever make it through successfully.
Okay, so if a compound does survive that gauntlet, the company applies for an investigational new drug, IND license, from the regulatory authorities like the FDA.
That's the next step, yes. If the preclinical data looks acceptable, the regulators grant the IND, and then, finally, clinical trials in humans can begin, conducted under those strict good clinical practice, GCP standards we discussed earlier.
Now, the sources mention a relatively newer strategy called Phase 0. What is that?
Yes. Phase 0, sometimes called microdosing studies, is a non-mandatory, relatively recent approach. It's an exploratory step that can happen very early, sometimes even before extensive chronic animal toxicity testing is complete.
Microdosing. Tiny doses.
Extremely tiny, yes. Subpharmacological doses, usually no more than 100 micrograms total, are given to a very small number of human volunteers, maybe 10 or 15 people.
What's the point if the dose is too low to have an effect?
The purpose isn't to look for efficacy or safety. It's purely to determine the drug's basic human pharmacokinetics, absorption, distribution, metabolism, excretion very early on, using highly sensitive analytical instruments like accelerated mass spectrometry, AMS, that can detect these minuscule amounts.
Why do that so early?
The idea is to get an early read on whether the drug behaves in humans as expected based on animal studies. If a compound shows really poor absorption or unexpectedly rapid elimination in humans right at Phase 0, the company can decide to drop it immediately before investing huge amounts of time and money in extensive, long-term animal toxicity tests and larger human trials. It's an early filtration step to weed out candidates with unsuitable human PK.
Makes sense. Save resources. Okay, then we get to the traditional Phase I trials.
Right. Phase I is the first administration of the drug to humans at doses expected to be pharmacologically active. Typically, the studies involve a small number of subjects, usually 20 to 80, and they are often healthy volunteers, not patients.
And the main goal of Phase I.
The primary focus is safety and tolerability. Researchers carefully escalate the dose to determine the maximum tolerated dose range, MTD. They also collect detailed information on the drug's human pharmacokinetics, PK, at different dose levels, and look closely for any immediate adverse effects. These studies are usually open label, meaning everyone knows who's getting the drug.
Okay, Phase I is about safety in healthy people. What about Phase II?
Phase 2 is where the focus shifts to efficacy, and the studies involve patients who actually have the target disease or condition, typically maybe 100 to 500 patients.
So does it work?
That's the main question, yes. Phase 2 aims to provide the first real evidence that the drug has the desired therapeutic effect in patients. It's also crucial for dose ranging, exploring different doses to find the optimal balance between efficacy and side effects, and identifying the ceiling effect, the dose above which you don't get much additional benefit. Phase 2 trials are usually randomized and controlled, often against a placebo.
And if it fails in Phase II?
If efficacy is not clearly demonstrated in Phase 2, the drug is usually abandoned at this point. This is a major hurdle where many drugs fail.
Okay. If it succeeds in Phase II, it moves to Phase III. This is the big one.
This is generally the largest, most complex, and most expensive phase, yes. Phase 3 trials are the confirmation stage. They involve a much larger number of patients, typically from 500 up to 3,000 or even more, often conducted across multiple centers, sometimes in different countries, multicentric.
And the design?
They are almost always randomized, double-blind, controlled trials. The main goal is to definitively establish the drug's clinical benefit, its efficacy and safety, in a larger, more diverse patient population, and critically, to compare its value against the current standard therapy if one exists.
And success here leads to?
Success in Phase 3 provides the core evidence needed to submit a new drug application, NDA, or equivalent regulatory submission to agencies like the FDA or EMA seeking permission to market the drug.
But even after approval, the research isn't over, right? There's Phase 4.
Correct. Phase 4 studies, also known as post-marketing surveillance, begin after the drug has been approved and is available to the public.
Why keep studying it?
Because now the drug is being used by a much larger and more varied population than was studied in the pre-marketing trials. Phase 4 is absolutely critical for detecting uncommon or rare adverse effects, long-term side effects that might take years to appear, and previously unsuspected drug interactions that just weren't seen in the smaller, more controlled Phase I3 trial.
And studying special groups.
Yes, Phase 4 also provides the opportunity and is often required by regulators to conduct specific studies in populations that were typically excluded from earlier trials, like children, the elderly, or pregnant women, to get better data on safety and dosing in these important groups. It's ongoing safety monitoring, essentially.
Okay, let's try to tie a lot of this together now. Pathology, altering drug effects, individualized dosing issues, adverse effects, using that problem-directed study example the sources provided, furosemide in hepatic cirrhosis. Can you walk us through that scenario?
Sure, it's a really illustrative case. Imagine a 65-year-old patient with advanced liver cirrhosis who presents with gross ascites, massive fluid buildup in the abdomen. The clinician, wanting to get rid of the fluid quickly, prescribes a high dose of the powerful diuretic furosemide Lasix. The drug works as intended, inducing a brisk diuresis, lots of urine output.
Before? No good.
Technically, yes, the fluid starts coming off. But then on the third day of treatment, the patient becomes increasingly weak, confused, and disoriented. Laboratory tests are done, and they reveal dangerously low serum potassium levels, hypokalemia, and a high blood pH, indicating metabolic alkalosis.
So the intervention, while achieving its immediate goal of diuresis, actually precipitated a catastrophic outcome, right? Hepatic encephalopathy.
Exactly. The patient slipped into hepatic encephalopathy. And the mechanism is a perfect demonstration of how underlying pathological states fundamentally alter drug outcomes.
Explain the link. How did the furosemide trigger it?
Well, the aggressive diuresis caused by the high-dose furosemide led directly to significant losses of potassium and hydrogen ions in the urine. This resulted in severe hypokalemia and the metabolic alkalosis, the high blood pH. Now, the research confirms that this alkaline state in the body favors the chemical conversion of the relatively non-toxic ammonium ion, which is TXNH4 plus duller, into the highly lipid-soluble toxic gas ammonia, TXNH33.
And ammonia is bad for the brain.
Very bad, especially when the liver isn't working properly. The cirrhotic liver is already failing to detoxify ammonia effectively from the gut. So this new surge of ammonia, generated because of the diuretic-induced alkalosis, readily crosses the already somewhat leaky blood-brain barrier in these patients.
Causing the confusion and coma?
Precisely. It causes severe mental derangement, confusion, flapping tremor, asterixis, and ultimately coma, the hallmarks of hepatic encephalopathy.
So the dose of furosemide might have been correct just for the goal of diuresis.
Yes, purely from a diuretic standpoint, it achieved the effect.
But the consequence of that dose was entirely altered, dangerously so, by the patient's underlying liver pathology.
Exactly. It led to a catastrophic result that likely could have been avoided by using a more gradual diuretic approach, perhaps combining furosemide with a potassium-sparing diuretic, or by monitoring and aggressively correcting the resulting electrolyte imbalances, particularly the hypokalemia and alkalosis. It just perfectly highlights the critical need for constant vigilance regarding the patient's entire clinical picture of that milieu. #tag outro. So we have covered a huge amount of ground today, a truly comprehensive deep dive. We've journeyed from the most fundamental definitions of dose and dosing strategies all the way through to the most rigorous standards of evidence-based drug development and evaluation. And I think the consistent theme, the core takeaway, is that achieving true professional mastery in using medicines hinges on recognizing, understanding, and actively accounting for the profound physiological variations between individuals.
Yeah, differences in size, the huge impact of age, both infancy and old age, with their unique PK and PD shifts, the way specific diseases like liver or kidney failure fundamentally compromise drug elimination and sensitivity.
And looking forward, the rapidly evolving understanding of how our individual genetic makeup dictates so much of our drug response.
So what does this all mean, really? It feels like the complexity of individual patient variability is just vast. Relying solely on population averages on that standard dose seems increasingly inadequate, almost crude in some ways.
It often is. And the tools of pharmacogenomics, while still evolving for routine practice, are becoming sharper, faster, and potentially cheaper all the time.
Which brings us to that final provocative thought.
Right. The simple biological fact remains that your core genotype, your DNA sequence, doesn't change over your lifetime. So unlike checking blood pressure or kidney function tests, which need repeating, genotyping only really needs to be done once. So given the accelerating pace of genomic technology and the now well-established critical impact of known genetic polymorphisms on the efficacy and toxicity of many common important drugs, we really have to ask, what is the true timeline for pharmacogenomic screening to move beyond being a specialized, often costly application? When will it become a universal, routine, maybe even point-of-care step, integrated into every single patient's initial prescribing protocol?
That feels like the ultimate hurdle for truly personalized medicine, doesn't it?
Yeah.
Making that genetic information readily available and actionable for every prescriber, for every patient right from the start.
It is arguably the biggest hurdle and perhaps the biggest promise for the future of optimizing drug therapy.
A fascinating and incredibly essential thought for us all to consider as we move forward. Thank you for joining us for this deep dive. We sincerely hope you feel thoroughly informed and equipped with that elite-level knowledge we strive for.
Until next time.